these talks over the last year or last few years really, and they all sort of vary from place to place, different slides and that. So a lot of the time, I don't know what slide's coming up next, and it sort of adds to the excitement, I have to say, for me anyway. So the next slide is, talking about abiotic stress, okay? There's two different types of stress really that our plants have to put up with. There's abiotic, which means it's caused by a non-living organism. Now, normally I would talk a fair bit about this, but today we're just going to briefly mention it. So it's non-living stresses caused usually by too little or too much of these. I'm pointing here. I know you can't see me pointing, but too much or too little of these factors here on the left of the screen. Too much water, too little water, temperature too high or too low. And the thing about that is, when plants are abiotically stressed, they also respond with a whole range of biochemical responses. And a lot of the time, these are very similar, if not identical, to the responses plants have when they're being attacked by a pathogen. And that's interesting. For example, there is plenty of research showing, say, low temperature stress, where, say, turf grasses have been exposed to 0 degrees centigrade for a number of weeks. Plants would respond to those low temperatures. But these plants then following on will be much more resistant to say microdochium infection, because the defenses are already in place. But anyway, today it's mostly biotic stress we want to look at, and this is caused, of course, by a living organism. So this can be other plants in the area. It could be by disease agents or predators, but really we want to talk about pathogens, which are basically living organisms which cause problems to our turf grasses. They can affect the visual and the playing quality. And biotic stresses, of course, can be caused by things like insects. Actually, in my own experience, a biotic stress I had to put up with for a number of years at the Royal Curragh were sheep. They'd be grazing the course and they would be eating very tightly the grasses, and the organic output from the sheep would be causing problems. So that was a sort of a biotic stress I had to put up with. But insects, fungi, bacteria, and viruses, which I don't really want to mention too much of, all cause biotic stresses. But again, today we're really going to look at fungi. But you might wonder, why the hell do these microorganisms, why do they infect our plants? What's the attraction? And of course, really, they're just like us. These microorganisms, such as fungi, they just want to eat and drink and reproduce their species. And of course, plants are a very attractive source of nutrients for them. And of course, not all these microorganisms are pathogenic. The fact that the vast majority of these organisms are beneficial, and in fact, we wouldn't have plant growth really without microorganisms. They're there to recycle, distribute nutrients, and another picture of a virus there, sorry. But they can be very beneficial to plants, so they're not all bad. So you have things like mycorrhizal fungi, you have rhizobium, which is nitrogen fixing bacteria. But as I say, plants require microorganisms, and it's only a very small percentage that will infect or cause problems with particular species. And these have evolved over the millennia to attack their own host species. So the vast majority are beneficial. But what we're looking at, actually, that's just a nice graphic here of a sort of a fungus infecting a plant. You can see, I find my little pointer thing. Hope you can see it. It's this one that's jiggling around. So you can see, say, a fungal spore here germinating and growing down. In this case, it's entering the plant via the stomata. So it goes through the plant and it's there entering the cells, extracting nutrients. So that's what it wants to do. It just wants to get fed and reproduce, basically. So again, we're just going to concentrate on the couple of pathogens that cause diseases in our turf grasses. But crucially, and this is the main part of the talk today, is that we want to see how turf grasses and plants actually respond with these defense mechanisms. And you might wonder, why should we bother about, learning about or inquiring about these defense responses? Why are they so important to, say, to greenkeepers and turfgrass managers? Well, here we go. Legislative restrictions over the last few years. We all know these fungicides here on the left. They're all restricted. They're all gone. We do have some fungicides still. And actually, by the way, I talk a lot about looking at different ways to reduce disease and that, and you might think I'm anti-fungicide, but I'm not. Obviously, if you have fungicides, use them because they do a great job and they're safe once you use the label rates and that. But we'll have to look at ways to enhance whatever fungicides we have and look at ways to at alternative ways to reduce disease. And I've talked a lot over the last couple of years about looking at these different factors. As you can see here, this is the disease pyramid, where we have, there we go, here's my little marker. Now there's 4 factors there which are interacting to affect disease levels. Now you can look at environmental factors. I often talk about cultural practices and nutritional inputs, how we can use these to reduce disease. But today we're going to, this talk is basically on the pathogen, the host plant, and the time factor, these interactions here. And when I mean time, I mean we need to look at how we can speed up basically how the infected plant can recognize it's being infected and speed up how we can respond. And by doing that, we can actually then have less disease incidents, hopefully. That's one way that we're going to look at to reduce disease in the future and of course at present. So just a quick run through of a sort of a typical infection process. And this very is a sort of generic infection process for any sort of fungal pathogen. It could be Mycodochium, Anthracnose, it could be Claridia, any of them. So the source of the infection, the inoculum, would be in the soil root zone areas. And basically when environmental conditions are correct for that particular pathogen, it will start to infect the host plant. And then you'll find the hyphae, the mycelium, will grow onto the plant. It will enter the plant. Some, as with Mycodochium, will enter via stomatal penetration. Others form what's called aprosoria, which are structures which burst through the outer layer of the leaf, the cuticle. So they will physically break through the plant, into the plant. When they're in, they have to enter break down the cell walls, and they have to extract nutrients, and then they'll exit and produce reproductive structures, and then these are spread and the whole process reoccurs. But the key thing today is that infected plants respond with these induced defense mechanisms. Now, sometimes I'm often asked whether plants actually have an immune system. Now, when I say often asked, I'm not actually often asked. I don't be walking down through Lidl and meeting people and saying, "Have you seen the specials today? And by the way, do plants have an immune system?" No, but sometimes when you're talking about defense mechanisms and plants, people say, "Do plants have an immune system?" Like we do. Like we're mammals. If we become infected with any sort of like measles or smallpox, you can be inoculated against these, and our bodies then produce antibodies to fight off, and for future infections, in most cases we're immune. Plants don't have an immune system as such, but they've evolved a very complex array of structural and chemical and protein-based defenses, and these will first of all detect the invading pathogen, and they will try to stop them, and then also then signal to other parts of the plant system, and then these can become less susceptible to future infections. And this here somewhere, this here is a sort of a grid or a mechanism for defense. Now you could take a screenshot of that because it's showing a lot of stuff here, okay? And if you look at, let's say, the left-hand side here where my red marker is moving around, you can see you have, first of all, as part of the defense mechanism, you have this what's called passive or constitutive. And these are defenses that are in place all the time during normal growth, and they include structural defenses and also biochemical defenses. And then you have the more interesting ones, I think, which have these, what's called over here on the right, you have these active or induced defenses. And these occur upon elicitation of infection. And again, these induced defenses can be structural or biochemical. And we're going to go through a few of these now. So the first thing is the first layer of defense is actually in place all the time, these constitutive defenses. And if you see here in this diagram, you can see they're actually physical barriers. And they're things, fairly obvious things really, like you have a waxy cuticle, you have cell walls. These are all barriers to stop penetration. Stomata, the size and shape can affect that. Various other structures are formed like callus. You have chemical barriers as well. Just click on further here. I'm just looking at my watch here. I want to make sure we're staying on time. So, you know, the thickness of the cuticle, which is present all the time, and the cell walls are very important structures to keep out most pathogens. Some plants have evolved physiologically. For example, if you think of fescue grasses, which are tightly rolled leaves, compared to, say, POA, the fescue leaves are going to be a lot drier just from the actual shape of them, which means there's less chance of the fungal conidia growing on them, less water on it, less moisture, less growth. Some plants have developed very thick layers of cuticle, such as bark. I was trying to think of some grasses that have bark, but I couldn't. But that's just to give you an idea of the physical barriers that plants have in place to prevent infection. And you might think, so how are they getting infected if they have these barriers? Okay, well, most of them are keeping out the pathogens, but a few for each species have evolved ways around these barriers and will get in. As well as those physical barriers, which are in place all the time, we have some chemical compounds which are also produced as part of normal growth. And these compounds are called things like phytoanticipins. Again, they're molecular compounds which act to attack directly the fungi or whatever's invading them. And on the whole, they're fairly successful. Again, there's always a few successful pathogens that have evolved a way around these. So they're in place all the time, these, and on the whole, they work fairly well. But it's these, they're the structural and the chemical ones that are in place, but it's these here that are more interesting. I think these are induced. And these occur when the pathogen invades and the plant recognizes it. And for these induced defenses, it involves a lot of stuff actually. So it involves things which we're going to mention, things like elicitors, the hypersensitive response, the programmed cell death, reactive oxygen species, pathogenesis-related proteins, phytoalexins, phenolic compounds, systemic acquired resistance, induced systemic resistance. So I'm just going to briefly don't let that put you off now. This is quite straightforward enough once we get through it. So that they're all the stuff that's involved in this induced defense mechanisms in plants. Salicylic acid is another key one, which is actually a form of aspirin, which is a signaling compound in plants as well. So inducible defenses, right? This is a sort of a timeline. When I say a timeline, it doesn't go from A to B to C to D. So the first line of defense could be going on while at the same time other parts of the defense will be initiating. And it's pretty a lot of interaction going on in this. But what we do first is we look at this very first line of defense, and it involves, first of all, recognition of by the plant that there is a problem. Something has to set the alarm off. And then once it recognizes it's being attacked, various signals will occur at the molecular level. There's ions, ion fluxes it's called. Ions will move from the molecules, from the plant cells out, and other ones will move in. And you get what's called an oxidative burst and cell death. So just to expand on that there a little bit, the first thing you have to note is that elicitors are required. So this is a compound which stimulates the response of plant defense. And of course, like I said, it could be an abiotic stress or biotic, but elicitors will recognize there's something going on. And once the alarm goes off, the plant will start to respond. So this is a sort of a diagram of what's going on. I'll just get my little red marker. So this is the cell, and here's the pathogen here, which is going to break in. It's breaking in. And elicitors can be things like some of the molecules can break away from the pathogen, and these will be picked up and recognized by the plant. Or, you know, when the pathogen is actually breaking down cell walls, particles of the plant cell walls will be floating around basically, and again picked up. So these are elicitors which are picked up, they set the alarm off, and once that happens then various signaling compounds are produced, defense chemicals kick in, and responses begin to happen. So the very first response is what's called the oxidative burst. So what is that? Well, various types of oxygen species are produced, things like superoxide, hydroxyl, but the main oxidative burst component is actually hydrogen peroxide. And this leads to what's called the hypersensitive response. So basically what happens here is the elicitor is picked up, it sets off the alarm, these reactive oxygen species are produced, hydrogen peroxide in particular, and this hydrogen peroxide acts either as a signaling compound, which starts off other defenses, which leads to the cells surrounding the infection site to die, which is trying to contain the infection. It'll also have a direct effect on the pathogen itself. So this is what I'm talking about here with this programmed cell death. This is one of the main first responses to the hypersensitive response. So if you see over this leaf here on the right, this is you can see these infection sites here. And what's happening here is the hypersensitive response has kicked in, and the cells in the surrounding area have actually died. And the whole idea is for the infection to be contained, because the pathogen is out to get nutrients, and if the cells are dead, it's sort of contained and sort of starved out. That's the idea there. So while the programmed cell death is going on, that's just a grammatical feature of what's going on. There's up this top right, top left here is the cell, the fungal pathogens coming in, it breaks into it here, into the cell. Starting to extract nutrients, the signals are going out, the hypersensitive response is beginning up here, and then the cells around that site are actually dead. So while that's going on, I say hydrogen peroxide is key to that. And as part of our research a few years back, we use what's called fluorescence microscopy, where we use various stains which react with hydrogen peroxide. And you can see here on the right, these are infected, I think they're POA leaves, so there could be creeping bent, I'm not sure offhand at the moment, but the creeping they're turfgrass leaves which are infected with microdocium. And you can see these blue areas here are the infection sites. And this blue is actually the burst of hydrogen peroxide as part of this response. So hydrogen peroxide plays some key roles in this first response. It induces defense-related genes. So while the container is going on, while the containment is going on, it's sending out signals around, and other cells within the area will actually be strengthened as well. So it causes lignification of cells just outside the infection area. It also can have a direct effect, hydrogen peroxide, on invading pathogens. I just listened there, the cat was meowing there, I'm not sure what's wrong with him. But anyway, so hydrogen peroxide, or this oxidative burst, is crucial in all plants for various stress responses, but it also can be quite dangerous in plants because it can also damage the plant if it goes out of control. It's a bit like a nuclear reaction, which can be very beneficial, but it has to be contained. So the plant also has to produce what's called antioxidants as well. So it's a balancing act, really. But anyway, hydrogen peroxide and this programmed cell death is key to the first line of defense in plants. Again, that's just another photo of an infected poa leaf. There you can see this burst of blue at all the infection sites. We also actually, as part of the research, we actually extracted hydrogen peroxide from infected plants, and we were able to measure and see exactly how much was being generated. And that's just showing there 72 hours post-infection. You can see where it peaked there about 12 hours post-infection. Anyway, that was the sort of the first line of defense there, this first box here on the left. But while that containment is going on within the infection area itself, as I say, the signals are going out, and the plant will start to fight back and start producing, start to strengthen cell walls in other areas, but also start producing what's called secondary metabolites, which are sort of antimicrobial compounds, which will directly attack the the pathogen. So just to go back over these antimicrobial compounds that are being produced by the plants. So a lot of these are called what's called pathogenic related proteins, PR proteins, and they're produced under stress. They're widely distributed in plants under pathogen attack, and they have a number of functions. First of all, they will directly attack microbes and pathogens. That also, some of them are used to strengthen cell walls and various other parts of the plant. An example of these PR proteins would be what's called chitinase. Now, fungi cell walls contain mostly chitin. So these PR proteins, some of them are, as I say, are chitinase. And this means that when they come in contact with the fungal cell walls, they will actually break down the chitin. And I can tell you the fungi don't like that. So that's one way the plant fights back. Another PR protein or antimicrobial compound is what's called PHYTOALEXINS. Again, these are compounds which are produced following infection and elicitation, and they can be very toxic to attacking organisms. For example, they can puncture the cell walls of the pathogen, of the fungus. They can affect and disrupt the growth and metabolism of the fungi and prevent the reproduction of it. They have numerous functions, actually, and it's been shown in various research that by removing or stopping the production of phytoalexins, well, then the plants become much more susceptible to infection. So they are a crucial response for plants to infection. So they have to occur for the plant to be able to any chance of fighting off infection. I have to say they can't be inherited, so you can't breed plants with these phytoalexins in place, unfortunately. So they have to occur during infection. And another thing I must say is that phytoalexin is pretty much a generic term. And it covers, because each plant species seems to produce its own form of these antimicrobial compounds. For example, if you see in this thing on the right here, this resveratrol here is produced mostly in grapes. This one here, pistatin, is in peas actually. And this one down here is mostly in oats. So each species produces its own unique form of phytoalexin. But they're antimicrobial compounds, and they're very important for reducing attack. So just a very quick review of early responses at pathogen attack. Obviously, the main thing is elicitation. The plant has to recognize it's being attacked. Then it produces this HR, this response, and cell death in the surrounding areas of the infection. Other cell walls in the area are reinforced by lignin, which strengthens them, so that the pathogen finds it harder to break through. Then there's various synthesis of these PR proteins and accumulation of phytoalexins at infection sites. And the thing about all these compounds, these PR proteins, phytoalexins, these signaling compounds, these lignin, they're all actually what's called phenolic compounds. So they can be grouped in together as phenolic compounds. And what exactly are these? Phenolics, okay? Well, you know, plants actually produce pretty much thousands of various types of phenolic compounds. When I say they're secondary metabolites, in other words, main metabolites in plants are those produced during normal growth and reproduction. Secondary metabolites are produced in times of stress or as a response to various problems. And phenolics are what's called, they're hydroxyl, they're actually what's called an aromatic ring molecule. So they're ring shaped, which basically means they're very strong actually compared to other shapes. But what we need to know is that they're produced in numerous types of compounds in plants, phenolics, and a lot of them are very keen and very important for defense mechanisms, a whole range of defense mechanisms. They have a lot of functions in plants, these phenolics actually. That's just some of them. For example, they form ultraviolet screens, so they help protect against heavy sunlight. They produce a lot of signaling compounds for various processes. Pigmentation, anthocyanins are phenolic compounds. So a lot of the pigmentation molecules in plants are phenolics. They have a role in plant growth, and of course in plant defense, they're key as well. And in plant defense, phenolics are part of these phytoanticipins, they're phytoalexins. They're part of the cell wall strengthening, like lignification. They actually have feeding deterrents. Now, like I said, with abiotic stress I mentioned earlier, for example, if an insect is eating a plant, the plant won't sit back and take it. It'll respond as a complex chain of events occurs. But at the end of it, if you remember that the insect, for example, is there to extract and take in proteins from the plant, and the insect will then, once it takes the proteins in, will use what's called proteinase to break down those, and the plant will respond after being chewed on, for example, by producing what's called proteinase inhibitors, which goes into the insect when it's eating it, and it will help to stop the protein in the insect being broken down. So, it's a very complex area. But anyway, what we're interested in, of course, is the defense compounds produced upon infection. And actually, this is something we did as well a few years back. This is Poa annua over 3 years, and we looked at the total phenolic compounds in these control plants, which were not infected, and we compared it with levels of phenolics in the infected plants in the red. And as you can see, over the 3 years, the infected plants had much higher levels of these phenolic compounds, which helps support the fact that they are defense related and they are produced in response to infection. That's just another fluorescent microscopy image of an infection site. You can see the blue hyphae infecting the plant, the turfgrass. And the yellowy green color is this burst of what's called phytoalexins at the infection site. So anyway, back to this sort of diagram of infection. We've seen the very first line of defense, the containment. We've seen that plants start to fight back within the affected area, producing all these antimicrobial compounds and cell strengthening. But it's this long-term systemic resistance that we're really interested in, because this can lead to the plant becoming more resistant to infection, to future infections, in fact. So there's 2 types of this sort of whole plant systemic response. You can have what's called, I better sit up before I fall off the chair altogether. You can have what's called systemic acquired resistance, or induced systemic resistance. And basically, they're pretty similar, really. They rely on biological or chemical factors to induce these responses, and then systems, internal messenger systems in the plant will travel through, and it will lead to increased resistance throughout the whole plant, basically. So what's the difference between the two? Well, this is systemic acquired resistance, SAR, and this relies on, I was going with my laser there, I'm pointing with my finger, that wouldn't work, so I use this. So this systemic acquired resistance allows for this here, where my red is pointing, this is a plant being attacked by a pathogen. The first lines of defenses are going on, but while that's going on, signals are sent out to the rest of the plant, and this allows for production of all this sort of these defense compounds throughout the rest of the plant. So in future infections, then the plant is primed and ready to attack or to respond to attack. With ISR, it's much the same, except you can induce this without a pathogen. In fact, you can use various factors such as beneficial organisms in the root, or you can apply treatments to the plant, which again induces defense responses and signaling compounds. A lot of the time, there's different signaling pathways induced in these, but at the end of the day, these systemic responses will lead to a plant which is much more able to respond to infection later on. That's just another thing of the ISR and SAR. So, oh, that's the same one, sorry, I put that up twice. So anyway, the systemic resistance is very important for us, because if we can enhance or use it to our advantage, we can sort of reduce disease in plants. So it's a whole plant response. There's several pathways involved in this, and it allows plants to respond more quickly to future infections. And for this sort of whole plant response, salicylic acid is one of the key compounds used for signaling. Again, salicylic acid is a phenolic compound as well, as a matter of fact. Okay, so I'll just put this up first. So basically what happens is you have the infection going on in your plant over here. Salicylic acid then will be produced in other parts of the plant, and these defense compounds are then in place, ready to respond. So I'm just going to give an overview of this before we go to other parts, because I'm looking at my clock here to the right, just make sure I want to stay on time. So host defenses can be in place all the time, or they can be induced, and they can be structural or biochemical in nature. The induced ones, which are the most interesting ones, rely on elicitors to recognize they're being attacked and set off the alarms. You have the oxidative bursts, you have this production of hydrogen peroxide, your hypersensitive response, cell death trying to contain the infection. You have other areas then, the cells are going to be strengthened through lignification. Then the signaling compounds going out while that's going on to other parts of the plant. Plants begin to fight back, first of all in the infected area, and then throughout the whole plant. So these PR proteins and phytoalexins are produced. Salicylic acid is traveling through the rest of the plant, inducing things like this long-term systemic resistance, such as SAR and ISR. So that's sort of the brief outline of defense responses in plants and turf grasses. And of course, the big question is, can these actually be stimulated or enhanced? Can we actually use these to our advantage? And yeah, we're doing it all the time, or quite a lot of the time anyway, because we can use what's called defense enhancers. So these are treatments which will try to control diseases by priming these defense mechanisms prior to infection, ensuring the plant is more able to respond. And there's things like silica, civitas, chitin, salicylic acid, harpin, and phosphate, obviously. So these are all elements and compounds that are available throughout the UK and Ireland in most companies I think will have these in their products these days, and you can include them into a program. And will they be effective is the thing. So how do they work? Well, silica, for one, is is one thing, for example. And this is an example of defense enhancement through the physical strengthening of the cuticle and the cell walls. So this is it here on the left. As you're looking at it, you can see a fungi breaking into the cells here on the infected plant. And by applying a layer of silica, which will, well, actually applying silica, it'll form a layer around the cell walls, is the theory. And it will add an extra barrier. So it should stop infection or slow it down. Silica, or silicon as the Americans call it, it can physically stop or slow down the infection. As a matter of interest though, it also has a biochemical effect on a lot of fungi as well. But this is one I've used. If you're looking for a silica product, look for something with a 10 to 20% VW or WV amount. It's pretty easy to apply. 3 litres per hectare should be enough for that, but it will have an effect. Now, before I go any further, of course, any of the things I'm just going to mention won't control disease on their own. Obviously, you can put them into a program with other compounds and that, but you should be looking at cultural and nutritional inputs as well, obviously. But this is silica down from Lawrence Datnoff. He used it to have an effect on grey leaf spot on a warm season grass, and you can see that it reduced disease on its own. And again, with another compound, with chlorothalonil, it's very effective. So silica in your program can strengthen physically your plant defense mechanisms. I'm just going to mention Civitas. This is a product from Canada. It's used a lot in the States and Canada. And this is a great example of induced systemic resistance. So it's a mineral oil which is applied to plants prior to infection, and it induces these response mechanisms in plants and is very effective at reducing disease levels. This is it on snow mold or mycodochium. The bottom line is the control level. And you can see using the Civitas here, where I'm jiggling around, good reduction on disease levels. This is the effect on anthracnose. Again, control up on top. Civitas, again, very effective. And you can see in the picture here how effective it is. And this is primarily from enhancing the plant's defense mechanisms prior to infection. This is chitin. I'm going to mention this. Chitin, or chitosan, is a derivative of it. As I say, it's part of the fungal cell walls. It forms the fungal cell walls, chitin does. And a lot of research in agriculture in the last couple of years, because it can act as an elicitor. So is it effective though? That's the question. Well, how does it work? Well, you know, as I said regarding elicitors, if fragments of the fungal cell walls break away, these chitin fragments are picked up by the plant, and this starts the response in the plants. So by applying chitin or chitosan, the plant thinks it's actually being infected, and it will start these infection processes. That's one there I'm trying at the moment. It's 10% chitosan. Again, you know, I have to do some more work with that, but it should, if you're if you include it into your program, it will have some effect on enhancing your defense mechanisms. This is, as you can see here, the top line is an untreated control. The fungicide, very effective, and the chitosan also had an effect just by priming the defense mechanisms again before infection occurs. Salicylic acid, I mentioned that just a minute ago. It's a key signaling compound in plants for the systemic acquired resistance. And applying salicylic acid before infection, again, it's priming the defense mechanisms. And you know, there's research from Penn State on perennial ryegrass. They applied a form of salicylic acid prior to infection, and it had a very significant reduction on an infection grey leaf spot, actually, on perennial ryegrass, again by priming these defenses. Harpin is another elicitor, which again, this is available throughout the UK, and again, applying that will actually prime the plant as part of a program, of course. I'm going to mention phosphate, as I always do. Phosphate, form of phosphorus, taken up in plants as phosphate. Phosphite is PO3, phosphate is PO4. So phosphite can't be used as a form of phosphorus nutrition, but very effective over the years at reducing diseases such as Pythium and Phytophthora. There's some research with anthracnose and microdopium in cereals. We did a lot of research over 4 seasons, which we published in 2012, 8 years ago. Yeah, so we trialed phosphite for microdopium suppression, and we got very good results. Reduced microdopium by 50% sequentially. How was it done? Well, we looked at numerous ways in which it reduced disease, but one we looked at was regarding enhancement of defense responses. As I say, we looked at 2 areas that I've already mentioned: hydrogen peroxide generation and phenolic compounds. Various processes we used, and this is hydrogen peroxide accumulation in Cretan bent and POA. But what we found from our fluorescence microscopy work was that you can see the infection sites here and this burst of blue, which indicates hydrogen peroxide and this oxidative burst. But what we found was that, pre-treatment with phosphite actually enhanced the speed of this oxidative burst, this hydrogen peroxide generation. So it was actually helping with this first line of defense. So we looked at all these, if you remember, these pathogenesis-related proteins, these phytoalexins, these are all phenolic compounds. And again, we looked at, I showed you this a minute ago, we looked at the production of phenolic compounds in turf grasses. And we looked at pre-treating non-infected plants for 6 months. And after the 6 months, pre-treated with phosphite, after 6 months of sequential treatments, we found these levels of these defense compounds, these phenolics, were much elevated in the phosphite-treated plants. And this carried on through into the greenhouse treatments. This is POA annua, which we had in the greenhouse. And we looked at the level of phenolic compounds in the infected plants, microdopium infected plants. And you can see the phosphite-treated plants produce much higher levels of these defense compounds much more rapidly. And as I say, that carried through because we had much lower levels of infection again. So the phosphite was actually enhancing these defense processes. So research supporting defense enhancement. Well, we saw that 2008 to 2014 one, I just mentioned that. But 2 I'm just going to briefly mention are the anthracnose suppression in 2018 and microdopium 2019. So this is anthracnose, and you can see in this chart, sorry, just give me a thing, Banner Max, very low levels of anthracnose, control pretty high. The phosphite here, treatment, good suppression, and this nutritional cocktail, which included NP and K, but also included compounds such as included phosphite, which was enhancing the defenses. But the nutritional cocktail also included things like calcium, manganese, and zinc, which didn't go into today, but these are all crucial elements for production of various defense compounds. We also included salicylic acid in that cocktail, and we also had silica. So that the combination there of various nutrients, but also defense enhancers, was very effective at reducing anthracnose. This is the microdopium trial, which we finished last year. Control here, high levels of microdopium. The propiconazole, when we had it, was very effective, of course. The phosphite on its own, again, reduced microdopium. But this combination here included, apart from phosphite, also included calcium, silica, and salicylic acid. And again, a good reduction of microdopium there through defense enhancement. So we're getting near the end of this, and looking at the clock here, I better get a move on. The biotic and abiotic stress are pretty similar. Our first line of defense is this physical barrier, such as waxes and cutin. These are in place all the time. But we have a secondary arsenal of inducible defenses. And crucial for this, of course, is our elicitors, which recognize the attack, and the hypersensitive response, which looks to stop, contain the infection. But after that, then we have these induced responses which travel right through the plant and allow much more systemic or whole plant defense. Salicylic acid is key for that. There's a whole lot of phenolic compounds produced, and then we can actually use defense activation to prime or enhance these. Again, if we look at our pyramid, it's this pathogen host plant time interaction. So priming and these defenses is important. It will work. All these can have play part in your program, and they will work to help reduce disease. And in conclusion, hey, plant responses to either biotic or abiotic stress involve very complex interactions and signaling, much more involved than what we can get into today, obviously. But priming, enhancing these responses can be key to our disease suppression. So that's me on Twitter. This is the view from my office during the summer, and that's it. We have a good few moments for questions, so hopefully we have some here.
Yes, we do, John. Thank you for that. We've got a two-part question to start with. How reliable are elicitors? Would you expect all species and cultivars to show the same response?
Sorry, what's the second part?
Would you expect all species and cultivars to show the same response?
Well, it's a great question, and unless you actually do the full research on it, you can't give a really proper answer. So I would expect most species would have some effect on them, all right, but it's going to vary, I think. Really, unless you do a lot of research into them bit by bit, species by species, you can't give a definitive answer, unfortunately. But I think that they are available at the moment, these enhancers, and I think it certainly would be worth putting them into your program and evaluating yourself how the effects are having as well. That's all I can say on that at the minute.
We've got some other questions, John. If plants can induce defences against infection, why do they wait until they're infected before they fight back?
That's another very good question, I have to say. Yeah, when you see, it's like us, right? All these defense mechanisms, these phenolic compounds, all these signaling compounds, they all require resources. There's no such thing as a free lunch, either with plants. So they have to have the resources to produce these compounds. And they're not going to do that unless they really need to, because their main function is to grow, metabolize, and reproduce. So, when they are challenged by pathogens, they will produce these. But again, for turf grasses, you have to consider that you have to be on a balanced nutritional program so that they actually have the resources to produce them.
Okay, we have more questions. Can the use of defence activators replace the use of fungicides to control disease?
Well, on their own, probably not. But I would say, defence activators in conjunction with good greenkeeping, good cultural practices, good use of nutritional inputs, possible biological inputs like compost teas could be a factor. If you combine all them together, you will, I think, be able to reduce disease levels to unacceptable. I mean, the last two trials I just showed there, we reduced anthracnose without fungicides to the same level of incidence as the fungicide inputs. So it was there's a lot of work still can be done, but it's very encouraging. You will get good results just from various practices such as these.
Another question, how soon before expected disease pressure should we incorporate defence measures?
Well, again, it's, how long is a piece of string really, but you would need, as you're treating these plants, they have to have time and, like I said, resources to produce these compounds. So I know certainly with phosphate, which I've done a lot of work with, I know pre-treatment with phosphate will take 2 to 3 weeks, we say, for these various compounds to be kicked in and respond to the treatment. So I would imagine a couple of weeks, but I can't say anything definite for each of these, you know, but say it's a gray area. So it's, but you certainly would need, it's not going to be an instant response in plants. So you would need a little bit of time prior to infection to get them in.
And last question I've got here, what other methods could be used to suppress disease?
Well, I think I sort of touched on them there now. Apart from, you know, if you have fungicides, use them.
Yeah, we've got another.
You have another?
Yeah, I've got another question.
Okay.
Shall I go with that?
Yeah, do that, yeah.
Can you reduce disease outbreaks by increasing good fungi and bacteria levels, reducing the space for pathogens, competitive exclusion? Hopefully I read that okay.
Yeah, well, you can actually. I mean, I've seen research using compost, which is what you're talking about. Is you're bringing in competitive microorganisms which will outcompete the pathogenic fungi. So using compost, there's solid data there to support that. It will reduce disease, and you know, the whole thing about our root zones is, if they're devoid of, or if they're short on various microbial activity, well, then the pathogenic fungi are going to have, basically going to have a field day and get in. So a healthy soil, a good balanced soil is, or root zone, is crucial, all right. And again, I have to say, with regards to inputs of like compost teas and biological inputs, a lot more research is required here, and there's not that much in it. So people want to start working on that and start producing papers and solid data in that area.
We have another question.
Okay.
What was the zinc and manganese product you used for reducing anthracnose, and can you get it in the UK?
Yeah, well, that sounds like something that came from a sales guy. He wants me to mention. It's probably one of his, maybe. No, yeah. The zinc and manganese was part of the, actually, it's a program we put together for that for the anthracnose trial. And it's in various products that are available now in the UK. And, you know, I'm not here to advertise, okay? But it's in the States. It's actually known as the Dempsey program, I believe. I was only told about that the other day. It's a list of various products, 5 products together, and they're marketed as the Dempsey product because it's the products I used on that trial. So yes, the zinc and manganese products, yeah, they are available. As a matter of fact, most companies, if you look, will have those elements in some of their products. So I'm not going to favor any, you know, on this webinar. I think it's only fair that we stay neutral. That's okay.
The questions are loading in now. Do you
dear.
have an opinion on the charcoal-based products being used in disease defences?
Well, the straight answer is no, because I don't know anything about them, to be honest with you. And, you know, when I'm doing talks, if I don't know the answer, I'll just say it. So I actually don't.
And we have a confirmation from a reputable source that it is the Dempsey program.
Ah, dear.
John, that concludes the questions that we've got here. So I just want to thank you for taking time to do this webinar, and thank everybody for attending. John, if you'd like to put the CPD code up on that last slide,
well, I forgot about that. Yeah, there's the CPD now for everyone.
There you go. That's the code for everybody to claim your CPD credits. You can join us again next Tuesday, when you'll be relieved to hear that Sammy will be back, and she'll be joined by Natalie from Exact to cover the updated furlough system and people returning to work, how to manage those situations. Hope you've enjoyed today, and thank you all very much. Bye.
Bye.